So hi, hello, and welcome again. Microbehunter here, Oliver here, and welcome to another Saturday microscopy evening. It's actually a live stream or not. Well, why do I say or not? Because this video has been pre-recorded again. I've been doing this already a few times in the past because today I have got again a time conflict. And this means that the chat is available for you. You can ask questions, you can communicate with each other, but unfortunately I will not be available for answering questions. I will try to answer those questions that you might have or those comments that you might have maybe in an upcoming live stream. So this means that today's video is also going to be a little bit shorter than the usual 1.5 hours, but I want to keep the pattern every Saturday. So even if I personally don't have time, then I would still like to make a video available so that those of you who expect something on the Saturday that you're not disappointed. Today I'm going to show you a technique how to slice this fir leaf from a fir tree, from a Christmas tree, and into thin cross sections. You've already seen probably before my microtome over here. I'll be using this as well, but I would like to show you also a different technique rather, one that I've basically just been using now just very recently, and I would like to first show you how this works with the microtome, and then I would like to actually show you the different technique as well. So microtomes are devices that allow you to make very thin cross sections. And how do you now place this specimen into the microtome here? You need a carrot or another holder to essentially hold the specimen. You can use a hole punch. Where is my hole punch here? To punch out the carrot. What I've done is I've used a knife to kind of trim it down a little bit, because the instructions are very clear about this. It's very important that the carrot fits very tightly into the hole here, right? So I'm going to turn this down. First, I hope I'm getting the correct direction. So I'm turning this down, and I managed to fit it in tightly. And what I have to do next is I have to basically put this leaf that I would like to cut into the carrot. How do I do that? I'm going to use now this plate here as a on the bottom, and I'm going to make a cut in here all the way into the center. Okay, and I'm going to now put this in here, obviously. So, and I somehow have to feed it in here, more or less straight. Here you can see it. Okay, it should go out on the other side as well. Otherwise, here we go. Right. Okay. Here we go. And then the whole thing here. Well, let's do it like this. Yeah. The whole thing then goes into here, but I just see that it's standing out here, so I'll do it like this, So, and I will now force it into the hole here. So the instructions mentioned that it has to fit very tightly, okay, and I will tell you then also why this is important, okay. So, and here we go, right, okay. So. And then I can essentially start to make my first cut, and there is a very sharp knife that I have here, and then I'm also going to show you some of the limitations of this method. Well, of course I could clamp it to my table here as well, but I'm not going to do that now. And then I have to essentially try to give it a clean cut, and when you kind of touch the surface here, you're going to notice that it actually goes down a little bit. Maybe a quarter of a millimeter, right? It's not completely flush with the surface. Okay, I'm just saying, right? So I'm now going to turn it. Now, into which direction do I have to turn it? I always forget. Okay, it's going further down. Now I'm turning it up. And it's slowly coming up, and then I can try to make another cut. Okay, you see. It's not quite as easy here. Okay, I'll try to make another cut. Better not cut myself. You can see. Okay, here we go. And if you look very carefully, you might be able to find it. I have to refocus this here. Wherever it is. Here it is. Okay, and then what you have to do is you have to put it into a little bit of water so that it doesn't dry out. So let's put this away again. Okay. So we turn this again a few times, and yeah, better not cut my finger. Okay, yeah, okay, yeah. You kind of get the idea. Here it is again. Yeah, and yeah. So this is basically yeah, a possibility here. I'm going to get some slides now. And I'm going to put this now on a microscope slide, and we're going to have a look at it right away, because I actually wanted to show you the second technique. I need a little bit of water. Yeah, so I've got some water over here. Yeah, and here we go. I'm going to take my pipette. Okay, and I need some tweezers as well. And it would have been a good idea if I would have prepared my glasses, because I'm already a little bit have some issues yeah, with my vision. Let's put it in here. And this one over here as well. Yeah, and let's have now a look under the microscope. Or no, I'm not going to do that yet, because I would like to change the knife first. And let's see how this actually works with a razor blade, because I've got some razor blades here. Okay, so let's try to extend this again a little bit here. Okay, and now let's give it a try with a razor blade. So it's a fresh razor blade. We have to be extremely careful. And you notice already that the razor blades are quite flexible. So cutting it like this might not be a good idea. So what I prepared is I've got those little clips here. And those clips actually can be used not only as a protection on the side, but also as a support. So, and then let's give it a try now. Okay. Okay, let's give it a try. See, it glides over it. Yeah. So I have to extend it again a little bit. Yeah. Yeah. And see. And I'm going to show you what the problem is. Yeah, the problem is that it doesn't grip. Doesn't get a grip. So, and another problem that we have is you see that essentially, even if it's able to cut, it essentially there's not enough distance here, right? So what I have to try is I could try it like this, and it doesn't work. Okay, so this is essentially one of the problems that I had with those razor blades. However, there is a different technique with the razor blade. See, for example, this one here works again. Yeah, yeah, and here is the next cross section here. However, there is a different technique as well. So what I'm going to do now is I'm just going to put this here directly, under the microscope to have a look on how this actually. How thin actually the cross sections are. And then, yeah, here we go. And they appear to be already fairly thick. Yeah? So, but let's give it a little quality check, and I'm going to switch over now to my microscope. So here we go. Okay, so this is, I'm now using my 20x, my 20 times objective, and this is now one of those cross sections that I found. Yeah, you can see that it does look a little bit blurry, because there are several cell layers here that we see. That's just this thing here on the side is the so-called a resin canal. I'm going to show you over here, right? It's a resin canal. And we see the individual cells, and it's fairly green, of course, because of so many chloroplasts that we have, but it's also fairly dark. Yeah. So essentially, it's several cell layers thick, and I would like to now show you a different method, and maybe we're able to get some slightly thinner cross sections. As a matter of fact, yes, this is going to be something that I'll be showing you up. Now I bumped it into the microscope, into the slide here, but let me show you a couple of here, this one, for example. This one is not on the edge, but it's essentially also a top view. A piece of carrot we have here. Yeah. Let's have a look over here if there's some more. Yeah, here are two of them. Right. Let's go up a little bit with the magnification again. Yeah. So it's basically how they look like. This one over here as well. Yeah, it's fairly dark. Okay, so I'm going to now make some cuts directly with with a razor blade, and I'm going to now show you again how I did the preparation here. So we're going to go back to the desk view, okay? And one of the problems, before I do that, I will quickly explain to you one of the problems that I have generally with microtomes, and that is that when you cut across with a knife, then essentially because of the flexibility of the keratin of the specimen, it tends to lift it out a little bit, and therefore the cross sections are a little bit thicker than what they should be. So I just cut across it, and I can actually feel that the keratin is now a little bit lower. Yeah, so this means that when I turn it out again, I'm still not able to see, it slides over it still, right? Yeah, because it's too low, right? So you have to turn it out fairly much in order to get a decent cut, and then when you kind of feel again, then you might actually feel again that there is a little gap here. Here it's a little too high. Here it might be a little bit too low. And if the keratin is not really very tight in the hole, then essentially it's going to be lifted out quite a bit more. Okay, so this is a little bit the difficulty, when using microtomes, it could also be that maybe the keratin is not the best holder. Okay, maybe cork would also work. So there are different things that I could maybe try out. Okay, so but I would like to now show you a different technique as promised, and for that I need this new razor blade. It's really important that the razor blades are new, and I need a slide, and I'm going to get my slides over here. Okay, got a slide box. Oh yeah, here they are. Let me take a couple of slides out. What you need, see, these are the pieces of paper separating those slides here. And what you need is, of course, the specimen. So I'm going to, get another pine leaf. No, I don't need another one because over here I've got one, right? And you put it on here. And what you do is you put a little bit of tape over it. Okay, to fix it down, but not completely over it, because the place where you actually want to cut it, should be not taped. Let me refocus again here. Here we go. Okay, so that is number one, and then I'm going to go now under my stereo microscope, and what I'm going to do now is I'm going to simply make cuts by using a second slide as a guide, and then I'm going to simply slice the fir leaf under the microscope directly, and I'm going to also add a little bit of water. The reason is that it prevents the whole thing from drying out, because the cross sections are so thin that they very rapidly dry out. Okay, that's something I don't want. So I will be now switching over to my stereo microscope, and I'll show you now what I'm able to see there. So this here is now under the stereo microscope, and the first thing that I did is I fixed everything down with some sticky tape. You really want to make sure that there are not many tolerances. So I basically not only taped down the slide, but I also taped down the actual specimen itself. And essentially then I took a sharp razor blade and I started sectioning everything. I made this as a separate video already, so there is a little bit of a delay here. Okay, so the first cut I made a little bit thicker here. And what I did is with a fresh razor blade, I did apply quite a bit of pressure as well, okay? So I did not want to pass through the specimen multiple times, but actually just the first time. And then you can see that actually it was already cut. This one was a little bit too thin here, as a matter of fact, right? A little bit, it's a little bit of luck as well, because there is only a slide on the top, right? And this basically means that depending on the angle of the razor blade, it might cut not completely vertically, but a little bit diagonally as well. So I used quite a bit of pressure, and the flexibility of the blade then kind of made sure that it aligned properly with the slide on the top. Yeah, but look, it actually looks quite decent. Yeah. And again, there is water, a droplet of water here. I did try also to do the whole thing with those clips here, which I showed you already before. However, they were kind of bulky a little bit, and actually I found out that I do need the flexibility of the razor blade. And this kind of allows me to cut, yeah, basically with a little bit of tolerance, because with pressure, then the razor blade kind of aligned properly with the slide on the top. Yeah, so that's basically what I've done, and you can see that essentially the cross sections, they look pretty okay. Light green, so this actually means that yeah, they're semi-transparent. This means that the cross sections are fairly thin. Yeah, I could actually also maybe try to measure them out, the thickness using a micrometer slide. I didn't do that, but I think you get the point, right? And this is fairly quickly. And why do you need so many cross sections? Well, actually, I'd be happy with one good one. But if you make multiple cuts, then the chances are pretty simply better that at least one of them is going to be just right. Because there is, of course, a certain variation as well. Yeah, some of them are a little bit thicker, some of them are a little bit thinner, right? Yeah, statistical variation. Yeah. So, and then I tried the following. I tried to then not make a cross section, but also longitudinal section. And for this reason, because I'm right-handed, I had to move everything around again. Yeah, I had to change the angle. So you see that all of the places where I applied some sticky tape. Yeah. Because there is a little bit of wiggle room otherwise. Yeah, you see. Yeah, so I rotated the stage now, so that yeah, because the slide itself is still connected to the stage using tape. And then I basically have done the same thing now, but now this time in a different direction. And let's see how this actually works. Now I do not quite have as much area to press down on the leaf. Yeah, so I just basically decided to make a cut right through the center, and now the cuts are a little bit longer. Yeah, so and then I moved it again a little bit. Yeah, so yeah, this reminds me a little bit of a cucumber. Yeah, and another one. They're still all connected, so I could not yet see how thick these cross sections actually are. And another one. But as we're going to see in just a minute or so, that it actually worked surprisingly well. And now I simply cut across. Yep, and yeah, here we go. Yeah, I removed those that I do not need. Yeah, and they're still basically because of they're still sticking together. Yeah, because water is a little bit viscous, so also due to capillary action, it kind of sticks together. But then later on the microscope slide, when we add a little bit more water and a cover glass, we're able to see that it spreads apart quite nicely. Okay, so finished with the preparation now, and now we can actually take it to the next step where we're going to look at the whole thing under the microscope. So, okay, now let's make a slide, and let's try to check the quality of these cross sections here. Okay, very quickly, because as we can already see, they slowly start to dry out and they stick to the slide. I'm not quite sure about those here, if they're actually thin enough or not. We're going to see. I'm just going to simply drop everything directly on the water droplet here. And let's have a look. Difficult, of course, to pick up. Yeah, and so this one here is the last one I'm going to use. It's on the bottom side here. This actually seems to be a little bit more transparent than the other ones. So, so here we go. A little bit more water, maybe. So because if we add more water in the cover glass, then the sections stays spread out. Where is my cover glass? Here we go. I'm going to take a slightly larger cover. Oh, this one has a broken corner. There's a round one over here. So here we go. This one is dirty. Quickly wipe it. Actually, a little bit of dirt doesn't matter. Just more concerned about the quality of those cross sections here. Here we go. Okay, so now let's have a look. Where is my microscope view again? Yeah, let's take. So yeah, exactly. This one was from the microtome, and now this one here is. Here we go. This one here is from the one that I just made. Okay, and you tell me which cross sections are thinner. Okay, whole bunch of air bubbles. You see, of course, that it's significantly brighter. So this means the cross section already must be thinner. But let's have a look. What else do I have over here? Here again, another one. You know what? I'm going to close the condenser a teeny tiny bit. Yeah. Where are the others? Ah, I love it. So I think I'm going to start a little biology lesson now. Okay, so that's basically the one that where I have the longitudinal cross section. Look at this, isn't this great? Okay, I say biology lesson because this is actually part of high school biology. What I'm going to be talking about right now, it's the anatomy of a leaf. Okay. And yeah, so I think you get the idea that this one is slightly thicker. Look, the vessels on the inside here. But I think you get the idea that essentially this technique seems to work well, actually surprisingly well. What I'm going to do now, and that's where I'm going to start to explain a little bit the biology part here, just for those of you who are interested. Okay, a little bit of leaf anatomy, so bear with me. Let's go up a little bit with the magnification here as well. Look, that is now, look at the nice background. I've got DIC now. If you just want to see it in a regular bright field, I'm just going to take out this here and let's go down a little bit with the brightness. So that's now regular bright field. Okay, yeah. So yeah, maybe the magnification is a little bit too large. Yeah, so let's go down again. So I'm going to explain to you now the different parts of a leaf here, right? So first of all, question number one, where's the arrow? Yeah. Question number one, which surface side is the top surface and which one is the bottom surface? Is this here the top one or is this here the top one? Easy to see. The one where the cells are, stacked. Essentially like vertically like this, that's the top surface. And now I am actually going to go up a little bit with the magnification because I want to show you here. You've got a layer of cells here on the very top, that's called the upper epidermis, and you see that it's pretty transparent. It's not green. So it allows light to go through, and the upper epidermis produces a so-called a cuticle. A cuticle is made of wax and is a waxy layer on the surface which prevents the water from evaporating, right? And then you have over here a very dark green layer of these cells. You see how they are nicely arranged here. It's called the palisade mesophyll. Yeah, it's a palisade layer, and of course, because it's so green, a lot of photosynthesis is happening here, right? So it catches a lot of sunlight, and the upper epidermis is transparent because it allows sunlight to go through, right? And then you've got the upper epidermis, you've got the palisade mesophyll over here, right? And then look at this layer down here. Look. Yeah, there are cells all over the place, but there are also these white areas. These white areas are air spaces. Yeah, this is called the so-called spongy mesophyll. I think it shouldn't surprise you why it's called spongy, because it looks like a sponge. You have to reach over to the microscope mechanical stage now. Yeah, and essentially, there are air spaces in here because gas exchange happens. Oxygen is given off by photosynthesis. Carbon dioxide enters the cells. Then you have a lower epidermis down here. And I think maybe not possible to see now, but in the lower epidermis you also have so-called stomata, which are openings in the lower epidermis to allow air to go in and out. Yeah, because after all, the leaf requires carbon dioxide from the air to do photosynthesis. So you see, I think it's quite nice, I think, to make these cross sections directly, and it's actually not even very complicated. Yeah. So I think this is a very, very nice example, and if you just focus through the leaf here, you're able to see that actually, look, Yeah. Do you actually see over here that the air spaces, those white air spaces remain white? Yeah, those here. So this means that I think I'm yeah on a cellular level. Yeah. So there are no other cells that actually come into focus here, or not so many other cells. So maybe it's only one or two cell layers thick that I've made here. So you see that this technique under the stereo microscope of cutting the specimens actually produces surprisingly good results. Yeah, and it's easier than with a microtome. Again, the microtome might be useful for different specimens again, which I'm not able to cut like this. Yeah, maybe larger ones, maybe also specimens that are embedded in paraffin wax. But for direct leaf cuts, I think it's pretty nice. So this is if you want to find an excuse of actually buying yourself a stereo microscope, here is an excuse, right? Because it allows you to make these nice cross sections. I think that's really nice here. Look at the upper epidermis again over here, right? The palisade mesophyll, yeah. Over here, spongy layer, the spongy mesophyll. See the air spaces in between the individual cells. Yeah, so I'm quite happy with the result. But let's have a look now at some of the other cross sections in more detail. Yeah, these here, right? Of course, there's some dust and dirt here as well. Let's close the condenser again or go down with the light again. Yeah, yeah. And what we have is we have in the center here, we've got the vascular tissue. These are the cells that transport water up to the plant. There are a couple of air bubbles here as well. This large white area here is because it's the specimen ripped a little bit during the cutting process, and actually it's not even that big of a problem, I think, because if this happens, it actually shows that I'm probably already thinner than the thickness of a cell. Yeah. So this is quite nice as well. Okay, yeah. So let's have a look here. A couple of air bubbles. Yeah, and let's have a look, another look. Ah, yeah, here we are again. It's another cross section here. Yeah, and yeah, those here. There are two of them stacked on top of each other. Yeah. Let me remove the arrow again. I'm quite happy with the result. Just for comparison again, these are the ones. Yeah, that's a piece of carrot here. There's an orange one that's carrot, and yeah, and that cross section was actually so thick. Yeah, that essentially it's a top view. It's not a side view. It's a top view. So I have to actually flip it 90 degrees. I cannot do that now because the cover glass is on top. That's another carrot piece, and here should be exactly the ones that are just cut with a microtome. Yeah. And yeah, I think the images speak for themselves. In short, I am able to, without a microtome, I'm able to make significantly thinner cross sections. Yeah, and when I just discovered this a couple of days ago, I immediately thought I'm just going to share this with you. Yeah, this one here is again a little bit thicker. Yeah, but here this one is quite nice. Yeah, so that's basically this was basically the fir, the needle or the leaf of a fir tree. And what I've got now is I would like to show you now, or I would like to make some cross sections of some other plant stems in the same way, and then we're going to have a look how this looks like. So what we've got here is in alcohol, stems and leaves of a tulip flower. And why completely white? Because I exchanged the alcohol several times, because the alcohol removes the chlorophyll and then the liquid becomes green, and then I exchanged it several times, and now it's completely depigmented. Yeah, how old is this specimen? I think this must be already. I made this around I think just before the year 2000, so it's almost 25 years old. Yeah, and I've been storing it in here for 25 years now, and every time when I want to observe something, I take a small piece out and put the rest in again. Yeah, so today I'm going to do the same thing, and I would like to now also make very thin cross sections under the stereo microscope. However, we got to be aware that the alcohol is very volatile; it evaporates quickly, and when it evaporates, the specimen shrinks. So what I have to do later is I have to then take the specimen after I cut it. I have to put it directly into water. So let's give it a try, okay? And I'm going to let's remove the tape here again. I don't know if it's a good idea to use sticky tape here, because maybe the alcohol might react with the glue on the tape. I don't know. So let's remove this here. And I'm going to take out first one of those stems here. Okay. You cannot see it here. That's one of those. Yeah, the stem of a tulip flower. It's a monocot. Okay. This is significantly thicker. Okay, it's significantly thicker than the leaf of a fir tree. Let's give it a try in any case. So this here is now the stem of the tulip flower. Which I just have taken out of alcohol. And same story here. Basically, what it is, I pressed down on the stem. A little bit of the alcohol started to be squeezed out, but I wanted to make sure that, also this here does not completely dry out, because when it starts to dry out, it really starts to shrink. Now, the consistency of this stem, which was in alcohol, is a little bit different again, because plant material that was in alcohol starts to harden a little bit. It's a little bit harder, maybe even a little bit easier to cut as well, because it's not quite as soft. So the first cut was quite thick here. This one is a little bit irregular. It's kind of folded back onto itself here, right? Same story. The more cross sections that you cut, the higher the chance that you're able to find something that's kind of fine. Yeah, this thick one is still the one from before. Yeah, this one here is this. I just realized that I did not properly focus my stereo microscope. Yeah. So it's a little bit blurry, but I think you get the idea. As we're going to see then in just a minute or so, the cross sections were also at the size of individual cells. Yeah? So basically, it looked quite nice. Okay. Because of the hardening of the plant tissue by the alcohol, the cross sections were, even though they were so thin, were fairly stable as well, and that's actually a good thing. You have to be a little bit careful, obviously, because a razor blade has two edges that cut. So I have to be very careful that I'm not cutting myself. Yeah, you see, start, so again, with a lot of pressure, you can even see how the razor blade starts to bend. Now, the pressure is not so much downwards only, but also towards the slide itself, towards the top slide, because I wanted to have a proper guide. So I decided that after about, I don't know, 4 or 5 cross sections, here we can already see how it looks like. Okay, it's a little bit blurry here now. Finally, it's in focus. I think it looks okay, yeah? So basically, this one too will now go under the microscope, and then I'll be also doing a methylene blue staining. Looks very nice. So this here is now the stage of the stereo microscope, so that you see a little bit better how I've done that. The stem is now completely pressed flat, as you can see this. Actually makes it also easier to cut. Basically I just pressed down. Yeah. Oh my gosh, look how yellow my fingernail is. That's because of the carrot. That's the carotene is a very strong pigment. So I'm basically now trying to carefully separate the cross sections. I move over the slide a little bit, and then I again slice it. And here you can actually see how the razor blade actually bends. Yeah, because I want to make sure that it's a straight cut. So I applied a lot of pressure towards the guiding slide on the top. Yeah. Of course, you also want to have a slide on the bottom because otherwise you're going to just damage the stage of the stereo microscope, something that you don't want. Yeah, yeah. So, and yeah, that's pretty much it here. It actually worked surprisingly well, and the cross sections looked quite nice. Yeah, and then of course, before I forget, everything has to be immediately transferred into water because the alcohol evaporates very quickly, and then the specimens start to dehydrate and then really start to shrink and become fairly hard. And this is something I don't want, and for this reason, I transferred everything into one of those dishes that contained water and then was ready for mounting. Yeah, and the stem now completely pressed flat. Look, yeah, will now go back into the alcohol and it will then start to expand again, I think, yeah, after a couple of minutes. Let's now have a closer look at the cross sections that I just made. Okay, here they are, and let's make another slide, and then also I would like to do a little bit of methylene blue staining, but let's have first a look at the unstained sample. Yeah, here we go. A little bit of, I don't know. I'm just going to use the same water here. There might be still a bit of alcohol in there. It doesn't matter. Okay, and let me choose a very thin one, even if it's not a complete one. Here we go. Difficult to pick up. Here we go. You know what? I'm going to try another one as well. This one here might be a bit incomplete. Well, we're going to have a look. Okay, so where is my cover glass again? So, and this, I hope only one of them. It feels so thick. No, it's one. Yeah, you see, the water is not everywhere, but it's okay. So, where is my microscope? The microscope viewer here again. Yeah, let's put this away. And now let's have a look at the non-stained stem. And here, oh, it's way too bright. I need to go down. Here we go. Okay, and this is actually quite good because it allows us now to estimate indeed the thickness of the cross section if we're able to see one or two layers. Yeah. So you know what? I go down with the magnification first, so to get a better overview. So this is how it looks like. Typical monocot stem. With all of the vascular bundles for transporting the water and the nutrients are scattered throughout the stem. Okay, so that's a typical monocot arrangement. But let's have a look now at the other stem here. Here is the other one. It seems to be actually even a bit thinner. Okay, so let's go in with the magnification. Let's have a look on how thick the specimen actually is. Okay, let's focus. And one cell layer, it might be it's actually one cell layer thick, less than a cell layer. You see, I do not see the layer going out of focus and another layer coming in focus. Yeah, it's quite nice. Let's try DIC just for the fun of it, just for the color. Yeah, well, actually, I think the regular bright field is also fine. Let's go down here. So, and 20 times. So, yep, again. Yep, looks nice. So I'm going to now try to stain the whole thing now with methylene blue. I need to get my methylene blue solution. Here we go. Well, I'm going to try to apply it directly, a little bit careful that yeah, I'm not going to spill anything over the microscope. So it's called Löffler's solution. It's a ready made. So let's go here where it starts to contact. Yeah, it didn't mix yet, and we have to wait a little bit for the stain. Let's move everything here a little bit. Maybe we're going to increase the mixing this way. Yeah, you see, you know what? I'll take it out again and I'll apply the stain directly. So I think that's the easiest way. So let's put it on here. Refocus. Okay. I've got, here it is. Let's add a little more stain. Okay, and you see that, yeah, it only touches it here. So let's mix it a bit. So here we go. And all of the excess I would like to get removed. So it means I need a tissue paper. So, because I don't want to spill anything over. And let's see if those parts that turn blue, if we're able to see a slightly more contrast. Let me remove a little bit. You know what? I'm going to remove the excess stain here. So let's have a look. So into the microscope, and where is the scope view? Here we go. Yeah, it does look a bit, I don't know. Let's have a look here. This one here. Actually, it does look a little bit nicer. Yeah. Yeah, so I can imagine that if I were to cut it a little bit thicker, that maybe because I cut it too thin, the cell's contents were kind of removed. And yeah, here you see that some cells stain a little bit darker than others. So this might indicate a little bit something about the presence yeah, of still a little bit of cytoplasm. But actually this one here looks now quite, wow, this looks actually quite nice. This is bright field, by the way. Yeah, so I'm quite happy. I'm quite happy here with the results as well. Look at this. Yeah, here now the vascular tissues for transporting substances are here. Yeah. So maybe if I were to focus now, again, you see, yeah, it's only one cell layer thick. So I think quite a good result. Huh? Yeah, as I already mentioned before, I think that's a very good use of a stereo microscope. Yeah, and simply to make very thin cross sections and preparations. Okay. So what I would like to do now is I would also like to make now a cross section through a leaf. Okay, because in here I still have some leaves as well from the tulip plant, and let's yeah, do in the same way a cross section as well. So this here is now the leaf. On my stereo microscope, I added a little bit of alcohol, again, to prevent it from drying out. And cutting this specimen was not quite as easy as the other ones. Yeah, same procedure. I put a slide on top, I pressed down. I actually thought it's probably going to be easy because the leaf was already very flat. Yeah. But it turned out that it was actually very soft. Yeah, so this was actually the first cut to make sure that the edge is very smooth. And then, yeah, essentially I tried to move the slide and I basically cut it again, and I found out it was very soft. Yeah, see, look, it ripped quite easily. Yeah? So in that sense, it was a little bit unexpected because normally when you put plant tissues into alcohol, they harden, they become harder, and therefore should be easier to cut. But this one actually was surprisingly soft, and it was also ripping and tearing. So what I've therefore done is I therefore tried a different technique. So yeah, by pulling it, you're actually generating shear forces. Yeah, and yeah, this basically causes everything to rip. So what I then did is the following. I tried to cut it flat. So I simply tried to cut the whole length of the leaf at once by simply pressing down the razor blade and not pulling it so much. And this actually then did work out. Yeah. So yeah, here, yeah. You see, I was kind of pressing down, and then after I pressed down, then I pulled across. So I was already all the way down and then I simply separated the section by pulling across. Yeah, but I first pressed down and then I pulled across, and this actually did work. Okay, so there was actually much less damage done to the specimen this way, and then I transferred everything again into a little bit of water to remove the alcohol. Okay, and that's going to be the next step. Yeah, the leaf cross section started to dry very quickly. So yeah, I used again my tweezers to carefully pick up each one of them. Of course, I looked through the stereo microscope while I did that, and I simply transferred it, and you can actually see that it starts to move around a little bit occasionally. And the reason is because of the surface tension that the alcohol destroys, and this sometimes causes some of the specimens. This one not so much because already quite dry. But the alcohol destroys the surface tension. And then there's another problem that I found, and that is that when there was alcohol in there and when you put it into water, sometimes there are bubbles forming, and these bubbles are indeed something that can be a little bit disturbing. But yeah, so I'm going to show you how I now stained it with methylene blue again. So let's make another slide here, a little bit of water. I wonder if I should use methylene blue right away. I don't know. Let's have a look at it first like this. Let's put both of them now here. Of course, the orientation is very critical. I don't know into which direction it's now looking. I hope that it's not, the top-down view. I want to have the side view. So there is a little bit of an uncertainty here. Oh, this was bumping the camera here. Let's put a cover glass on top, and then let's have a look. Here we go. Okay, and I really hope that I'm looking at it from the right angle. So let's put it again under the microscope. Where is the microscope view? Let's take the old slide out. This is the old slide, and here this is now the leaf cross section. Here we go. That's overexposed. Lots of bubbles, usually because of the mixing of the alcohol, and the water. So this is how it looks like. I don't like the bubbles so much. Let's remove the arrow again. Let's zoom in a little bit more. Okay. Here again we see the different cell layers. It doesn't look so nice. It doesn't look so nice. A little bit a pity that, but still, it's fairly thin. Yeah, it's still a cell layer thick only, maybe 1 or 2 cell layers, but it's fairly dark. So what I'm going to do, just for the fun of it, let's do another staining with methylene blue, and then let's see how it looks like. So here we are again. I'm going to remove the cover glass right away. Why do I actually put it on here? Here it is. Both of them are still on here. A small drop of staining solution. And let's put this one on here. So, and let's give it another try. So, where is the microscope again? Here we go. This is of course way too... Here we go. Yep. The problem is again, the air bubbles make everything appear much darker than actually what it is. The reason being because of the differences of the refractive index. But actually now we are able to see slightly more details. So let's go up with... that's okay, I guess. Still, it's a little bit interesting that I'm not able... So that is... Now we can actually see how the methylene blue stain starts to... starts to enter the leaf. But it's still fairly dark. And when cutting it, I could also see that, yeah, or determine that it was not quite as stable as I expected it to be. But at least we can confirm that, yeah, I'm looking at it from the side. Yeah, we see here in the center again what appears to be a vascular bundle. It's basically again where xylem and phloem are, where water is transported. Yeah, yeah. So again, here the leaf surfaces and the individual cells in here, right? So in that sense, actually, it's quite fine. Let's go up yet further. So everything is in bright field now. Okay. Yeah, interesting. Yeah. Yeah, and I think the cross section might not be quite as thin as I expected, but if I want to focus here, I still only see one cell layer. Yeah, so yeah, it could be that. Yeah, you can actually see how the cells are cut open. It's actually quite interesting. Let me. Yeah, you can see this. Look at the cell here. Yeah, you see that it looks like over here it's sliced open here as well, right? And here as well. Yeah, so the cell contents has run out here as well. It's sliced open. Yeah. So these razor blades are really sharp. Okay, let's go down again a little bit with the magnification so that we have a better overview. Yeah, it's starting to look nicer. Okay. Yeah. You gotta give the stain a little bit of time to do its job, right? Yeah. Yeah. Here we go. That's here it's ripped a little bit. You might notice that the leaf starts to stain blue, even though the surrounding medium is not quite as blue. The reason is that the methylene blue reacts with certain chemicals in the cell, and therefore it has a higher affinity, yeah, to certain parts in the cell. Yeah. Okay, you can also see over here, that's also quite nice. Maybe you noticed that. Yeah, here it's fairly blue. Over here, yeah, it's less blue, and the reason is because the stain reacted away and has bound to the cells here, and that's why, yeah, essentially it's brighter over here, yeah, compared to over here. Okay, so that's basically yeah, that basically completes the cross section of the leaf. Yeah, okay, not so good here. Yeah. Here we go again. You see that it basically stains stronger on the outside. Yeah, I personally like the stem a little bit more than the leaves. Okay, but still, I think you get the idea, and you can also cut it. Yeah, even when it's not quite as stable. It was ripping quite easily, but if you carefully place the razor blade, in a flat way and not pulling it by not applying shear forces, then you're also able to cut it. Yeah, it's a question of trial and error. So, last specimen, I'm going to put another carrot under the microscope. Well, it's exactly the same carrot that I used as a holder at the beginning of this video, as a holder for the microtome, yeah, for making the cross sections. The carrot is already a little bit, how do you say, soft, because it's quite old already. But still it was quite easily cut. The cross sections were quite even. You can see here that, yeah, they're already a little bit transparent, which shows that the sections are quite thin. Yeah, and it basically made 3, 4, 5 cross sections, and then of course also put it under the microscope. Now the carrot itself, I found out, did not look quite as interesting as the other specimens, but still we're able to see a little bit the difference in the cell size, because as I'm going to show you in just a minute or so, there were several cell layers. It was actually 2 layers thick, even though I thought that the cross sections were very thin. So evidently the cells were significantly smaller. Yeah? So essentially this is what I've done, and then I put it directly on a microscope slide and then under the microscope. Yeah, I carefully picked them up with a tweezer and put them on a microscope slide just like before. And yeah, they were quite easily spread apart because the carrot was fairly thick. So there was, of course, no danger of it looking at it from the wrong side, flipping it over. I've had the problem with some of the leaves where essentially I looked at top and not from the side because the leaves were wider than essentially they were thick. So this was not a problem here. And then a little bit of water has to be added as well, and I could immediately see that the cross sections, they started to swell up a little bit because the cells already started to dry. Okay, here is a bad way of putting the cover glass on top. So I had to remove the cover glass again because one of those cross sections was actually not covered. Yeah, and that's basically it. And then everything went under the microscope. Yeah, okay, so that's basically now the carrot under the microscope. We can see that it's 2 cell layers thick. Now one cell layer is in focus, and then when I turn the focus knob, the second one comes into focus. Yeah, so the cross section, even though it was thin, is now actually 2 cell layers thick. So maybe the cells are a bit smaller. You know, that's quite well possible. Yeah, okay, I think I'm just going to call it a day for today. I'm going to call it quits. Yeah, of course, I would like to welcome you to post some comments below. And I hope to see you again soon on another Saturday live stream, maybe then in a real live stream, and not a pre-recorded one like this one over here. But I still hope that you kind of enjoyed it and that you learned something. All the best, happy microbe hunting as always, and see you around next time. Bye-bye.